Malden notes, we're up to the bottom of 1.30, and talking about different things in the cell briefly, and it says the nucleoid. So remember that in prokaryotes, we don't have a membrane-bound nucleus. So they still can call where the DNA is a nucleoid. But let's say we have a rod like E. coli, and of course it has DNA, and it's not in a nucleus. It says here a single circular chromosome. So one could draw it like this, but in fact it's said to be supercoiled, which is like if you took a rubber band and you keep twisting it and twisting it and twisting it, so it's kind of folds on itself again and again and again, and takes a very compact amount of space. So that's the DNA that's said to be supercoiled. Not just the double helix that we all know about, but like I said about the rubber band. Have it in the pocket. So rather than this, a better description would be something like this, where it's really tightly packed. Okay. It says about 4,000 genes. That varies among bacteria. On top of 1.31, you may remember that eukaryotes have proteins associated with them called histones. They're very conserved across species. The DNA is acidic. They're mostly basic, and they are complexed with the DNA. DNA is wrapped around the spools of histones, called nucleosomes, like beads on a string is the phrase that's often cited. Anyway, eukaryotes have them. Bacteria do not. Okay, bacteria no histones. Archaea have some. Again, there's something like but different. They have histone-like molecules. So archaea are different than bacteria and different from eukaryotes. Okay. Next, plasmids. So you may have heard of plasmids. Some, but not all bacteria, have some small circles of DNA. Not the main circle of DNA, not the main DNA, and I still probably drew this too big relative to this. And they are extra chromosomal, small, circular, have a few genes. Usually they don't have genes that are required for life. So the maintenance of these guys, their replication and division isn't so tightly controlled with the cell's replication and division, so that sometimes they're lost. But there's tricks one can do to make sure they're not lost. Anyway, so that's interesting, but there are a couple other things about the plasmids that sometimes they're transferred between bacteria, and remember I mentioned lateral gene transfer yesterday. Lateral is between peers, if you will. One bacteria gives some DNA to another, as compared to vertical gene transfer, which is, you inherit genes from your parents. Anyway, that happens naturally, but also something to keep in mind of, for as early as next week, we're going to work with them, that scientists a while ago figured, aha, maybe we can do something with this. That these plasmids, these small pieces of DNA, they replicate on their own, and maybe we can use these for our purposes. So scientists figured out ways you can break open the cell and separate this little piece of DNA from the big piece of DNA because of its size. And once you have that little piece of DNA out in your test tube in your lab, you can do tricks with it, like maybe add a gene to it. Like if you're interested in cloning a gene, like we'll say a practical gene, like the gene for insulin, that fights diabetes. So now you've taken this out, and I'm going to pull up the scale a little bit, and now there's some tricks, which we'll talk about next week, where you can add a gene to it, okay, outside outside the cell. And then there's also tricks where you can put it back in. And we're gonna do that. So, and now, see what you've done is you've inserted a gene from somewhere else into bacteria. And why is that good for anything? So remember how fast bacteria grow. You then grow these bacteria overnight. You have a ton of bacteria, and you've grown out the whole lot of this plasmid, so you have many copies of this gene. So that's one thing plasmids are good for. So plasmids are something that occurs naturally. Some interesting things about it in nature, lateral gene transfer, and also things that scientists have used them for. So that's, when you've heard the term biotechnologies, as using stuff that happens naturally and using it for our purposes. So let's see, in the Malden notes, it refers to them as genetic parasites. There's usually slower growth in cells carrying the plasmids. Sometimes they're unstable, they're not always common. Yeast also have plasmids. Okay? Some brief discussion of DNA there. Next, ribosomes. So again, you remember from general biology, ribosomes are the site of protein synthesis. They're structures made up of protein and RNA. I hope that's rings a bell with you. That's a pretty important thing, protein synthesis. They're often drawn in cartoons, something like this, with a large and a small subunit. Because in certain salt conditions, they can be separated. Both contain RNA. As it says in the Malden notes, the small subunits contains the ribosomal RNA, which Woese used to determine the sequence of, and thereby claim that archaea were a separate group. The large subunit contains the RNA enzyme peptidyltransferase that's involved in making protein, transferring the new amino acid onto the growing ribosome chain. Okay. So anyway, one reason I'm mentioning this here is, like some other things, prokaryotes do it a little differently than eukaryotes, and there's an odd-looking factoid, 3/4 of the way down to 1.31, that 70S is the total sedimentation rate for prokaryotic ribosomes and 80S for eukaryotes. So you're saying, what's the sedimentation rate? So first, the main thing is to know that they're different. The prokaryotic and eukaryotic ribosomes are different. Sedimentation rate, really, that's just what you have to know, that the prokaryotes are 70S, eukaryotes are 80S, but I'll explain this to you. One way of working with the subcellular organelles, things like ribosomes and mitochondria and such, is to separate them from each other by density. You can use a centrifuge. You know what a centrifuge is, right? The rapidly spins around. And there are centrifuges that can do things like spin down whole cells. That's pretty easy. But there are centrifuges that go with such a high speed, they generate such a high force, they can separate things that aren't that very dense, and they separate them from each other. And you can separate organelles from, say, mitochondria or lysosomes or ribosomes from each other. Okay? So what you do is you layer them on top of a media that's more than water. It's denser than water, and then when they spin, they go in this direction. Anyway, the S value is how dense they are and how much that sedimentation goes. And it's named after someone named Svedberg, who I guess was a pioneer in doing this. But the main point is prokaryotic ribosomes are 70S, eukaryotic are 80S. So they're a little bit different.
70 is less dense. 70 will be less dense than 80. Is that what you say?
Yeah.
And so to make things even more complex, archaea ribosomes are somewhat more similar to eukaryotes by sequence, but they're still 7